Okay, today should be pretty much review though. It's over nucleic acids, which we've talked about, but it's been a little while. So try to think of what is the monomer. Do you guys remember? What is the monomer? Say it out loud right now. Okay, you should have said maybe nucleotides. Hopefully that's what you said. Do you remember what the polymer is? What does it say it? Okay, should be RNA and DNA are forms of our polymer. Do you guys remember what the bond is called that bonds those nucleotides together? This one's a little trickier. Do you remember it? That's a phosphodiester bond. We talked about that, but it's been a while. The overall structure, so think about what elements are going to be in part of the structure for nucleic acids. Do you remember that? That's our carbon, hydrogen, oxygen, nitrogen, and phosphorus. Chomp. Okay, you also need to know the structure has a 5-carbon sugar, a phosphate group, and a nitrogen base or a nitrogenous base. Okay, and the function of our nucleic acids, we should maybe know that. Do you remember what that is? Okay, it should be to store our genetic information. So here's some pictures of those structures. If you didn't remember, we have our 5-carbon sugar. It can either be a deoxyribose or a ribose sugar. We have our phosphate group on the side, and then we have our nitrogenous base. So that's our adenine, thymine, guanine, cytosine, uracil. That's where those go. Okay, and then when we have multiple of them bonded together, the bond here in between them is our phosphodiester bond, and they make this overall shape, and then we have our double helix shape that we have. So here's another showing our phosphates being bonded on the side right here. Okay, so let's go over our differences between DNA and RNA. So one uses a ribose, the other uses a deoxyribose as the pentose or that 5-carbon 5-ring sugar. So we have our deoxyribose and our ribose. Do you guys see in the picture where they're different? Maybe pause the video and see if you can see the differences. Okay, so what's the difference? The difference, oops, did not pop up, is right here. We have an oxygen here on our ribose. Deoxy, no oxygen, is our deoxyribose. Okay, so we have the big difference is just in the sugar between our deoxy and our ribose. So that's going to be our DNA and our RNA. Okay, so the nitrogen base can also be different over here. So for DNA, do you remember what the nitrogen base options are? Okay, so they can be slightly different. For DNA, that's adenine, guanine, cytosine, and thymine. RNA, how is it different? Okay, for RNA, everything's the same except instead of thymine, we have uracil. Okay, so this is another difference between our DNA and our RNA. Okay, we also have a difference in the strandedness. So how many strands? Do you remember which one's double-stranded? So our DNA is double-stranded, and our RNA is going to be mostly single-stranded. We can have some that are double-stranded in regions, but sometimes it'll be single-stranded, then double-stranded, then single-stranded, and so it's not always double-stranded like DNA is. And what's this shape called for DNA? It's called a double helix, where it has those two, double because it's 2 strands, and helix because it's making that spiral. Okay, so think about what happens if we change the sequence of our nucleotides. So here we have a DNA sequence, and we change the sequence slightly. So a change in sequence is going to be a change in the coded information of that. So if they change the DNA, it's probably going to change the protein that it's being made or some other function of the DNA. Okay, and we've talked about our differences between our types of nitrogenous bases. We have purines and pyrimidines, but so this is review. Remember, our purines are adenine and guanine. Our pyrimidines are cytosine, thymine, and uracil. We can use this purine as 2 gold rings, gold for guanine, and as gold, so A for adenine, A and G. These are our double-ringed structures, so that's a purine. They're going to have 2 ringed structures, our adenine and our guanine. The other ones are only single rings that we have, and those are our pyrimidines. Okay, another thing we have talked about, but it's been a while, is that nucleic acids are directional. We have a end that has a 5' and an end that has a 3'. So we have 5' phosphate and our 3' hydroxyl. So we usually say that it's going to be synthesized, which we'll talk about next class, in the direction of 5' to 3'. And so I remember this because we also have our protein is directional. And if you remember with our protein, it goes in terminal to C terminal, and I remember that it rhymes. C rhymes with 3, so it goes into C and 5 to 3. Okay, you have them the other way, doesn't rhyme. So if that helps you, great. Otherwise, you're gonna have to just remember that it goes 5 to 3. Okay, so here's showing a structure of our nucleotide. So see, we have a 3' oxygen here, that's our 3' hydroxyl, and then our 5' is up here is where our phosphate is. So we're going to go from 5' to 3'. So when we're adding new nucleotides on, we're going to add it to that 3' end. So here's showing where they're all bonded together. So we have a 5' to a 3', then another 5' to a 3', 5' to 3', and it keeps going, so at one end we will have a 3' and the other end will have a 5'. Okay, so here we have a picture of our double-stranded DNA that's bonded together. What do you notice about the directionality, the direction of those strands? Okay, you probably should notice that it is called anti-parallel because on one side we have 5' and then 3', and the other side it's opposite, 3', 5'. So that one going one way, one going the other way is called anti-parallel, where they're going in opposite directions. Okay, so it's anti-parallel, 5' to 3 in one, 3 to 5 on the other. But it's still, when it's synthesized, going to be synthesized in the 5 to 3 direction. Okay, so it just depends on which way we're looking at it. So again, when we have our synthesized, it's going to always synthesize in that 5 to 3 direction. It's always synthesized 5 to 3. Okay, and then, so we have those phosphodiester bonds here in this backbone that are being formed, but then our two strands are held together by hydrogen bonds between these nitrogenous bases. So you might notice, what do you notice about there's differences in the hydrogen bonds between some of these nitrogenous bases. And you might notice that adenine and thymine, they each have 2 hydrogen bonds, but guanine and cytosine have 3 hydrogen bonds. Okay, so that means guanine and cytosine are actually going to be bonded together with a stronger bond than our adenine and thymine. And I remember that the curvy letters are stronger. I don't know why that helps me remember. If you have something else to help you remember, that's great. And so remember, what's this bond again when we have between our phosphate and our next one? That's our phosphodiester bond. That's where that phospho comes in. And it's a covalent bond, so it's a much stronger bond on our backbone than we have between our hydrogen bonds, which is not really a real type of bond, it's more of an attraction, kind of like magnets. And this is called our phosphate backbone on the sides where we have our phosphates. Okay, so now we want to think about the DNA in our cell. So in each cell, there's about 2 meters of DNA in one cell. That's a lot of DNA. And that would be if it was all stretched out, but it's not like that because we have to fit it inside a tiny little cell. So on average, a human has 37 trillion cells. So we need to fit that DNA into a very small, compact space. So how it does that is it's wrapping it very, very, very tightly. And so you can think about, I was going to have you guys get a piece of yarn and try this yourself, but you can think about if you use yarn or like your shoelace or something and you twist it and you twirl it and rotate it over and over again, you can get a little curl, right? And then if you keep twisting it even more and more and more, that curl then curls on itself. And then that curl, if you twist it even more tighter and tighter and tighter, that curl can then curl on itself. And so that's what's happening when we wrap it multiple times. It's the exact same thing that's happening with our DNA. We have that DNA, first it's wound around these proteins called histones, and then it coils, and then it's called supercoiling, where we're coiling a coil. And that's going to make it super, super, super condensed in our chromosomes so that it can actually fit inside of our cell. Okay, so it's wrapped around these proteins that are called histones, and then it's wrapped around it forms something called a nucleosome. It's just where we have histones kind of wrapped around each other. And then they stack on each other in the coils. And then those coils are getting stacked in our supercoils. So this is how we're going to make our chromosomes and be able to fit all of that DNA inside of our cells. Okay, so we can model it with yarn. If you would like to go find some yarn in my cabinet, you can. If you want to pause the video and go back, you can. If you go back to my big rolly cabinet in the back and open the left cabinet, there's a box and there should be yarn in there. But if you can't find it, it will be okay because I can't think of how else to tell you where the yarn is. Okay, we need to also talk about our differences between our prokaryotic and our eukaryotic DNA. So remember, prokaryotes are simple organisms. They don't have a nucleus. Our eukaryotes do have a nucleus, and their DNA is going to be slightly different as well because there's no nucleus in prokaryotes. Their DNA is packaged in a different way. It's still going to be double-stranded, but in this case for prokaryotes, they have circular DNA. So instead of our chromosome shapes like are in eukaryotes, we're going to have circle DNA shown here in this picture. Okay, they're only going to have one chromosome, unlike eukaryotes that have many, many chromosomes, like humans having 46 chromosomes. The DNA for prokaryotes just float around in the cytoplasm, and again, it's going to be supercoiled DNA, but there's no histones in this case. Eukaryotes, double-stranded, linear DNA, so in a line, multiple chromosomes usually in the nucleus, and it's going to be formed in chromatin, meaning it's packaged and condensed into those around those histone proteins. We can also have something in prokaryotes called plasmids, which will come into play later in the unit. And plasmids are small, little circular extra bits of DNA that still can get incorporated and their genes can still be expressed. It's just little extra DNA, and this is found a lot of times in prokaryotes. There are some eukaryotes that have plasmids too. I don't think humans do, but that's what a plasmid is. It's slightly different than just prokaryotic DNA, but just little extra bits of DNA that we can add.